Greetings everyone, and today we are going to discuss about nucleus. Nucleus was first discovered by Robert Brown in 1831. Robert Brown in 1831. Apart from which, when it comes to nucleus, it is basically the control center of the cell, brain of cell. It is the control center of the cell, brain of cell, and all of the metabolic activities depend on the action of uh, the material present inside the nucleus. Why? Because it contains the hereditary material, as we have previously known about it. What kind of hereditary material? DNA is present, which contains a special segment of DNA known as gene, in which the particular sequence occurs. So now we are going to discuss about the structure of structure. Now we are going to discuss about the structure of nucleus, what is which is in such a manner that here first and foremost structure of nucleus is in such a manner that it consists of four major parts. Eukaryotic nucleus consists of four major parts, uh, which includes nuclear, which includes nuclear membrane, which we also call as nuclear envelope, or we also call the nuclear membrane and nuclear envelope as karyotheca. After which there is nucleoplasm, nucleoplasm, and in that case, nucleoplasm basically a nucleoplasm is the fluid which is filled inside the nucleus, which is known as nucleoplasm. The nucleoplasm is also known as karyolymph. Apart from which, nucleoplasm is also known as karyolymph. Now let us discuss about another part, which is nucleolus, and fourthly, we are having chromatin. So previously discussing, now let us discuss some basic characteristics of animal and plant nucleus. Animal nucleus is found mostly in the center, and it is mostly found in the center, while in the plant cells it is found a bit away from the center because of the presence of a large central nucleus, a large central vacuole. In the presence, because of the presence of large central vacuole, there is the nucleus which is present a bit on the periphery. While when it comes to animal nucleus, it is present in the center, except there is an exception, except muscle fiber cells. In muscle fiber cells, the nucleus is not present in the center. In muscle fiber cells, a nucleus is not present in the center, and apart from this, muscle fibers involves are multinucleated. Why? Because they require high amount of functionality, they require high amount of commands in order to perform several metabolic activities, as they are involved in contraction and relaxation in order to perform locomotion within the body. So in that case, these are multinucleated, and that has another terminology that we can also call it as poenocytic. Poenocytic nucleus. Poenocytic cells are multinucleated cells, which involves examples of muscle fiber cells. Secondly, some cells are binucleated, which are known as binucleated cells or dikaryotic cells. Binucleated or dikaryotic cells. Apart from which, there are some cells which do not have a nucleus at all, such as cells including cells of RBCs, uh, as well as sieve tube cells, in plants. Apart from which, there is sieve tube cells, RBCs. There's example of sieve tube cells, RBCs, as well as xylem tracheates. These are some examples. Apart from which, when it comes to mononucleated, they are present in wide amount in our body. Now coming to the structure. First, let us discuss about the structure of nuclear membrane. Nuclear membrane is a double membrane. There is an inner and outer membrane. Inner membrane and outer membrane are uh, parallel to each other, which means they are not connected with each other, but at some intervals these are connected with each other at several junctions, at several points, which are known as nuclear pores. As you can see in the diagram as well, there is a presence of nuclear pores, and these nuclear pores basically are formed by virtue of a nucleoporin protein, by virtue of a protein known as nucleoporin protein, and that causes to fuse both of the inner and outer membrane at some intervals. Apart from which, the nucleoporin protein basically forms a channel through which the material can be taken inside the nucleus outside the nucleus, which means nucleocytoplasmic exchange of material can take place by the nucleoporin proteins nucleopore. So in that case, it is nucleopore which makes it successful. Apart from which, when it comes to our second part, we are having the space, as both of these are nuclear membrane is having inner and outer nuclear membrane. So both of these are parallel. As these are parallel, they are having a space in between them, which is known as perinuclear space, which is around 10 to 15 nanometer. Apart from which, uh, this perinuclear space 10 to 15 nanometer, which is having a fluid filled inside it. Apart from which, when it comes to other aspects, nuclear membrane is also known as karyotheca, also known as nuclear envelope, which is having several nuclear pores, which been mentioned. Sometimes small molecules can easily pass through the nuclear pores, but larger molecules require signaling permissions. Molecules require permission in terms of signaling that once large molecules try to enter inside the nucleus first gives command to the nuclear envelope to check whether the molecule is right or not. First, the signal goes to the nuclear material. I mean to say that the chromatin network. First of all, the message of a large molecule coming inside the nucleus is sent to the chromatin network, due to which the chromatin network sends a command to the nuclear envelope to whether the large molecule should be entered inside the cell or not. So in that case, this is the condition which is involved in nuclear whenever the large molecule is entering inside the nucleus. Apart from which, um, these are a few aspects, and the when it comes to second part, we are having nucleoplasm or karyolymph, As you can see in the diagram as well, this is the structure of nucleus. Here there is the nuclear membrane, nuclear envelope, on the outer side of which we are having on the outer nuclear membrane. That's for the structure of inner nuclear membrane. Outer nuclear membrane is continuous with the ER. Outer nuclear membrane continuous with ER is continuous with ER. As an outer nuclear membrane is continuous with ER, and at the outer nuclear membrane is continuous with ER and attached with ribosomes. It is attached with ribosomes. Apart from which, the outer nuclear membrane is also supposed to form the rough endoplasmic reticulum. So in that case, it is the function of outer nuclear membrane. Nucleoplasm is a fluid-filled space. It is a fluid which is present inside the nucleus, which is nucleoplasm. It consists of following enzymes, which includes DNA polymerase, RNA polymerase, as well as proteins. Are involved histone and non-histone protein, as well as metal ions such as magnesium ions are present in here. Apart from which, nucleotides are present here as well. Nucleotides are present. These are some materials present inside it. Apart from which, there is nucleolus. Nucleolus is also known as the brain of nucleus. Brain of cell is nucleus, and brain of nucleus is nucleolus. Brain of nucleus, and that is a membranous structure, which means it is not having a membrane. It consists of the following part. It consists of following part, which includes it is consisting of granular, peripheral granular, granular, peripheral granular area, which consists of peripheral granular area which is made up of ribosomal units. Apart from which, there is an internal core area, which is basically having internal core area, the major internal core area, which is made up of fibrillary proteins. There is an internal fibrillary area, fibrillary core, which is made up of R DNA and R RNA, which means ribosomal DNA and ribosomal RNA. Apart from which, the structure can be drawn in such a manner that here there is, or suppose this is nucleus. The core part is basically made up of rRNA and rDNA. This is the peripheral granular area, which is made up of ribosomal units, and the internal structure over here. This complete structure, the complete part, this complete highlighted part consisted consisted of chromatin, and it is a major site of construction. It is a major site of synthesis of ribosome. So the site of active ribosomal synthesis is nucleolus. And nucleolus, how does it forms ribosome? Basically, it is the site of synthesis of ribosomal RNA, which basically forms a ribosome, due to which there is protein synthesis occurring inside the cell. So whenever there is more demand of protein is equal to more number of nucleolus within the nucleus. So in that case, this is the case over here. Apart from which, when it comes to third part, there is chromatin. Chromatin is basically involved in the following parts. uh, Chromatin basically is a network. Chromatin network of DNA and histone proteins in such a manner that here the structure can be showcased in such a manner that here there is the nucleoplasm, endoplasmic reticulum continuous with the outer nuclear membrane, and nucleolus, which is present in the center. The number of nucleolus the number of nucleolus depend on the uh, individual feasibility of cell, depends on the individual feasibility of cell. Apart from which, nuclear envelope is there, nuclear pores are here. Apart from which, this is chromatin network. Here there is a presence of chromatin network. Now here, the chromatin network can be explained in such a manner that here it is uh, a network of DNA and histone protein. It is a network of DNA and histone proteins which involves it is a thin thread-like network. It is a network of thin thread-like structure of DNA and histone proteins, and is involved in and is involved in genetic activities of cell and is involved in genetic activities of cell and is involved in genetic activities of cell and is involved in the genetic activities of cell. The genetic material of cell is present inside the DNA in the form of gene, which is enclosed in the chromatin network inside alongside the histone protein. And this chromatin network basically uh, condenses. It is basically available in non-dividing cells. It is available in non-dividing cells, but it is available in non-dividing cells, and during dividing cells, during the M phase, mitotic phase, the chromatin network condenses. The chromatin network condenses and coils. The chromatin network condenses and coils. The chromatin network condenses and coils in order to form. The chromatin network condenses and coils to form. The chromatin network condenses and coils during the cell division in order to form chromosomes, and that chromosome basically uh, coils to form chromosomes, and that chromosome is a thick thread-like structure which can be easily viewed using the light microscope. And there are two chromatin present inside the nucleus. One chromatin is the heterochromatin, second chromatin is euchromatin. Heterochromatin is the chromatin which stained by once the nucleus is stained by any stain, any chemical, the chromatin network is divided into two parts. There are two major colors of chromatin. First chromatin is a heterochromatin, which is having dark color over it, which means the heterochromatin is darkly stained. On the second part, we are having the euchromatin. Euchromatin is lightly stained, which means it is having very light color whenever it is stained with a chemical. So in that case, there's a euchromatin and heterochromatin. In euchromatin, the gene is not tightly packed. In heterochromatin, the gene is quite tightly packed. That was a difference between both of these, apart from which this is chromosome, and this structure can be explained in such a manner that this is chromosome, this is a chromosome condensed, highly condensed and supercoiled form of the chromatin network, which consists of the different structure of proteins. This is the histone protein and DNA, which is wrapped within the histone protein. Now, why histone protein is there? Histone protein, for suppose we are having a DNA. For suppose this is nucleus, and within the nucleus, the DNA is present without any histone protein. Then what will happen? The DNA will start to the DNA will start to you know form a network with each other, due to which it will be just like a network of it would form form a congested network. It will form a congested network which would be difficult to separate at intervals, due to which in order to make it separable and individual, there is a presence of non-histone protein. There is a presence of non-histone protein inside it. Secondly, there is a presence of histone protein inside it. Mostly for provide for doing the packaging of DNA, the histone protein and non-histone protein is present inside it. That is why non-histone protein is also known as the packaging protein, also known as the packaging protein. And histone protein is basically a basic protein, while DNA is acidic, as it is a nucleic acid, so it is acidic. So in order to neutralize the DNA, histone protein being basic causes to neutralize the DNA's acidity by cancelling the DNA's acidity with its basicity, due to which the overall nucleus chromatin becomes neutral. Overall nucleus chromatin becomes neutral. So at that case, histone protein is there. It can also be caused by packaging protein, but we mostly consider non-histone protein to be doing that. So in that case, histone protein is a packaging protein, and non-histone is a packaging protein as well. This is all about this video. Hopefully, we meet